Okay, protection of the brain, the cranium, the meninges, the ventricles and cerebrospinal fluid, and the blood-brain barrier. The brain is very well protected. First of all, the cranium completely encloses the brain, and it's a hard, bony, I always say eggshell, but then it sounds like it would be like fragile, but eggshell around the brain. Then there are three meninges that protect the brain. And these are continuous with the meninges of the spinal cord that we just learned, the pia mater, arachnoid mater, and dura mater. And it's the same thing. The pia mater is closely attached to the surface of the brain. It carries blood vessels. The arachnoid mater is loosely attached to the pia mater by arachnoid trabeculae. The subarachnoid space between the pia mater and the arachnoid mater is full of cerebral spinal fluid. And then the arachnoid mater is closely attached to the dura mater. So far, all the same as we learned about the meninges in the spinal cord. Now, is something a little different. In the brain, the dura mater has two layers, a meningeal layer attached to the arachnoid mater and a periosteal layer attached to the skull. Between the two layers of the dura are dural sinuses, venous spaces that collect blood and return it from the brain to the body and this, systemic circulation of the body. The dura mater forms sheets that separate areas of the brain. The falx cerebri is found in the longitudinal fissure. It separates the cerebral hemispheres and encloses dural sinuses. The tentorium cerebelli is another fold of dura mater that is at right angles to the falx cerebri. It separates the cerebrum and cerebellum. Then there is a little tiny falx cerebelli that partly separates the cerebral hemispheres. Okay, a bit on the dural sinuses. Cerebral veins drain into bridging veins that drain into dural sinuses. A major dural sinus is the superior sagittal sinus. And here's a picture of the dural sinuses. There's the superior sagittal sinus, and there are many other little sinuses, and they all end up draining into the internal jugular vein, and from there into the superior vena cava, and then the heart. And I put pictures of all this for you to follow. I know that we haven't learned about the internal jugular vein, the superior vena cava, and the heart yet, but we will. Okay. This is a clinical correlation. Different types of bleeding within and around the brain are described in relation to meninges. So an epidural hematoma is between the skull and dura mater. A subdural hematoma is between the dura mater and arachnoid mater. A subarachnoid hemorrhage is between the arachnoid and pia mater, and an intracerebral hemorrhage is bleeding within the brain itself. Now, a word on the difference between a hemorrhage and a hematoma. A hemorrhage is active bleeding, and a hematoma is mostly clotted blood or completely clotted blood. Bleeding in a tight space is slower and the blood clots more quickly. So if we look at an epidural hematoma, because it's in the tight space between the dura mater and the skull, it pretty quickly, perhaps not quickly enough, but it more quickly than some of the other places that bleeding can occur, clots. Okay, because of where it is. A subdural hematoma, which is between the dura and the arachnoid mater, well, that's also a fairly tight space, so you'd also expect the bleeding to be stopped a little more quickly. But when we get into the space between the arachnoid and the pia mater, that's a space that usually just has cerebral spinal fluid in it. So if bleeding occurs in that space, you get a subarachnoid hemorrhage with active bleeding. And then if you have an intracerebral hemorrhage, the brain is quite soft, and bleeding can really extend for quite a bit within the brain without any tissues constricting it and causing it to clot and causing the bleeding to stop. Okay, so moving on to ventricles. Ventricles are cavities in the brain that are filled with cerebral spinal fluid. The brain has 4 ventricles. There are 2 lateral ventricles in the cerebrum. Then there is the 3rd ventricle in the diencephalon and the 4th ventricle posterior to the pons and superior medulla. Posterior to the pons and superior medulla. Okay. The lateral ventricles are quite large and they have a large C shape. They are not connected to each other. They are separated by a thin partition, the septum pellucidum, and they are connected to the 3rd ventricle, but not each other, by 2 interventricular foramen. The narrow 3rd ventricle in the diencephalon connects to the 4th ventricle in the brainstem by the cerebral aqueduct, and the 4th ventricle finally connects to the central canal of the spinal cord. Okay, a bit on cerebrospinal fluid. So cerebrospinal fluid helps to protect the brain. It surrounds it, supports it, and cushions it. The brain is very soft. It has the consistency of jello. So if there are any blows to the head, the brain can be thrown against the inside of the cranium, which would very much hurt the brain if it was not for the fact that it is cushioned by cerebral spinal fluid all around it. Sometimes it's hurt anyway if there's a severe enough blow to the head, but that's just unfortunate. So to give you an idea of how the cerebral spinal fluid helps the brain, I'm going to tell you the weight of the brain is usually about 3.3 pounds. But when the brain is suspended in cerebral spinal fluid, its effective weight is reduced to only 1.3 ounces. So it's just bobbing around inside there, almost weightlessly. The cerebral spinal fluid is produced by the choroid plexus, and choroid plexus is made of 2 elements, capillaries and surrounding ependymal cells, which, remember, we learned were one of the 4 support cells in the central nervous system. All of the ventricles contain choroid plexus that creates cerebral spinal fluid. Ependymal cells create cerebral spinal fluid by selectively transporting the water, oxygen, and nutrients in blood from the capillaries into the ventricles. The composition of cerebral spinal fluid is very different from that of blood. The oxygen and nutrients in the CSF diffuse into the interstitial space around the cells of the brain, where they can be taken up by the cells. Waste diffuse from the cells to the interstitial space into the CSF. The ependymal cells transport the waste and any substances that are not needed from the cerebral spinal fluid into the blood so that they can be gotten rid of. Okay, CSF flows between the 4 ventricles and the central canal. Then it flows out of the ventricles through holes in the 4th ventricle. There are 2 lateral apertures or holes and 1 median aperture. From the 4th ventricle, after it comes out of the holes, the CSF flows into the subarachnoid space around the brain and the spinal cord. From the subarachnoid space, CSF drains through arachnoid granulations into the dural sinuses. The arachnoid granulations pierce through the meningeal layer of the dura, and then from the arachnoid granulation, the cerebral spinal fluid joins the blood that is in the dural sinuses and is returned with the blood to the systemic blood, through the system of dural sinuses into the internal jugular vein and then into superior vena cava and then the heart. CSF is completely replaced every 8 to 12 hours, and the total volume is usually around 2 milliliters. So, not tons, but not an inconsiderable amount if you have an idea of what 200 milliliters looks like. Okay, the blood-brain barrier. The blood-brain barrier separates the blood from the brain. It's composed of the endothelial cells of capillaries, tight junctions between the cells, the basement membrane under the endothelium, pericytes, which surround capillaries and help support them. It's like the support cells for blood vessels. And then finally, astrocytes. And remember, when you drew your picture, you drew astrocytes that had processes that on their ends had tiles, and the tiles were used to create a sheath around capillaries. The blood-brain barrier is selective. It protects the brain from pathogens, toxins, and the immune system, but it actively transports glucose into the brain. If drugs are needed in the brain, the blood-brain barrier must be crossed, and that can be a problem. Finding something that is able to cross the blood-brain barrier and treat whatever problem the person is having in their brain can be a problem. Okay, the hypothalamus, pituitary gland, and pineal gland have a more permeable blood-brain barrier because these areas of the brain need to secrete hormones into the blood. So it is quite handy to have a more permeable blood-brain barrier in these areas so that the hormones can get into the blood. Okay, blood supply to the brain. The brain requires an enormous amount of oxygen, glucose, and other nutrients. And in order to bring blood to the brain, there are two major arteries. They are the common carotid artery, which then branches into an internal carotid artery that goes to the brain, and the vertebral artery. And you can look at that picture and appreciate those vessels, although we haven't learned them yet. Clinical correlation. An ischemic stroke is caused by a blood clot in a blood vessel that stops blood flow to an area of the brain. If blood flow to the brain is interrupted for 10 seconds, the person will lose consciousness. If it is interrupted for a few minutes, irreversible brain damage will begin. And that is why strokes must be treated very quickly. And there's a saying, time is brain. Because with each moment that passes, you're losing parts of the brain. Remember, the neurons do not regenerate. And so you've just lost part of your brain, which is like who you are, really, your brain. And if it's a big enough part, you are never the same. So that's why we have to treat these matters very, very seriously as strokes. I'm not laughing at strokes. I'm laughing at how I'm trying to explain, just laughing at myself. Okay, so